This video is going to be about single dominant epistasis. Epistasis is simply put the term for when one allele masks the expression of other alleles within a genotype. In single dominant epistasis, a dominant allele prevents all other alleles in the genotype from expressing phenotypes. I'm going to be using the coat color of gummy bears as an example. In order to understand this form of epistasis, we can look at the enzymatic pathway. Before looking at the pathway itself, a way that I conceptualize it is by thinking about it as a series of if-else statements, which I've shown here in the pseudocode. Doing a dihybrid cross with two heterogeneous white pairs, we obtain the following genotypes, shown here. Let's start with the genotype with a big A allele and a big B allele. If we follow the pseudocode, we find that if a big A allele is detected, the production of enzyme 1 is inhibited. If we look at the actual pathway itself here, we see that enzyme 1 is responsible for adding pigment to the white precursor phenotype, and the big A allele inhibits the production of this enzyme. Based on this, we know that single dominant epistasis is occurring, since the dominant A allele prevents the production of enzymes within the pathway. Since 12 bears in the progeny would be expected to have a big A allele, 12 out of 16 bears would be expected to be white. We can move on to the next part of the code, now that we have detected all genotypes with big A allele. Only genotypes without a big A allele remain. Let's say we detect the genotype with all recessive alleles. This would mean that enzyme 1 is produced, but not enzyme 2, since enzyme 2 requires a big B allele to be produced. That leaves us with an intermediate green pigment, which have had enzyme 1 added to the white precursor phenotype. Since only 1 out of 16 of the phenotypes in a dihybrid cross would have had recessive alleles, only 1 out of 16 bears would be expected to be green. Finally, the only genotypes remaining do not have a big A allele or two little b alleles. This genotype shown here has a big B allele that codes for the production of both enzyme 1 and enzyme 2, leaving us with red-colored end products. Since 3 out of 16 genotypes have two little A alleles and one big B allele, 3 out of 16 bears would be red. Moving on to this dihybrid cross, we can visualize the phenotypic ratios that this mode of epistasis yields. Here, we see that each genotype with the dominant A allele has white coloration, regardless of the other alleles paired with it. When we add up the total number of white-colored bears, we get 12. This has been contrasted with simple Mendelian inheritance, where there would be 9 bears with the big A, big B phenotype, which would have their own distinct phenotype, and 3 bears with the big A, little b alleles with their own distinct phenotype. The fact that both of these genotypes have the same phenotype tells us that the common allele between the two, big A, is responsible for masking the effect of the other alleles. The final phenotypic ratio here would be 12 to 3 to 1. In this cross, we will still see epistasis at work. We can observe that all the genotypes with big A allele end up being white, since this allele inhibits the production of pigment-producing enzyme 1. The other half of this progeny without a big A allele end up being red. This is a 1-to-1 ratio. In this cross, we do not see green pigment, since all genotypes have a dominant A allele or a dominant B allele.